Hello, this is Professor Torsen. This is the recorded lecture for Chapter 11. We begin our discussion of heredity by looking at the work of Gregor Mendel. Mendel was an Austrian monk who studied inheritance patterns in garden peas back in the 1800s. Before we go too much further, I want to introduce a few new glossary terms. This chapter is full of new glossary terms, and I suggest flashcards may be helpful for you guys learning these new glossary terms. If we look at this table at the bottom of slide 3, we see 7 different traits listed. So for example, one of the traits that Mendel studied was the color of flowers on the pea plants. Flower color is a gene, and that gene happens to have 2 different versions or alleles. There is an allele that dictates the plant have purple flowers, and there is an allele or version of the flower color gene that dictates the plant should have white flowers. On slide 4, we're looking at an outline of Mendel's experiment. He took a plant that had the phenotype of purple flowers and a plant that had the phenotype of white flowers and cross-fertilized them. This produced the next generation of plants called the F1 generation, and the F1 generation had the phenotype of all purple flowers. This F1 generation was allowed to self-fertilize and produced the next generation called F2. In the F2 generation, Mendel saw 75% of the plants had purple flowers. The phenotype was purple flowers. And 25% had the phenotype of white flowers. This was quite interesting as the white phenotype had disappeared in the F1 generation, but reappeared in the F2 generation. We can use Punnett squares to help us understand what was happening in this experiment. And here are 2 more glossary terms for you, homozygous and heterozygous. Mendel began his experiment by crossing a purple flowered plant and a white flowered plant. We can use the Punnett square to represent the genotypes of the parent plants shown on the left hand and the top side of the Punnett square. So here is the purple flowered plant, and we notice there are 2 alleles. Both are capital P. Capital P stands for the dominant allele, which is for purple flowers. Up top, we see the genotype for the white flowered parent plant, and it is 2 lowercase p alleles representing 2 recessive alleles for this gene. In the Punnett square, we see all 4 possible combinations of these alleles. In this quadrant, this offspring plant inherits a capital P or dominant allele from the purple parent and a lowercase or recessive allele from the white flowered parent. In this quadrant, again, this offspring inherits a capital P or dominant allele from the purple flowered parent plant and a recessive allele, lowercase p, from the white flowered plant. You can see for all 4 possible combinations, the only combination that will result from a homozygous dominant plant being crossed with a homozygous recessive plant are heterozygote individuals. All possible combinations result in organisms that have 1 dominant allele and 1 recessive allele. This is called heterozygotes. Notice the phenotype is of the dominant phenotype, purple flowers, because these individuals have one copy of the dominant allele. This illustrates Mendel's first law, his law of dominance. Essentially, we know that each parent contributes genetic information to the offspring. Mendel discovered that some traits were dominant, while others were recessive. These recessive traits appeared to skip generations, as we saw in this example from the previous slide, looking at purple and white flowered plants. There were no white flowered plants in the F1 generation. Mendel repeated this experiment with 7 different traits, as described here in this table on slide 7. For all 7 of these traits, he found very similar data. Essentially, 75% of the F2 generation had the dominant phenotype, and 25% of the F2 generation had the recessive phenotype. This can be seen in this table on slide 7. On slide 8, there is an image of a multi-hybrid Punnett square. It is possible to utilize Punnett squares to look at the potential outcome of a crossing of 2 individuals with respect to 2 different genes. However, we are not going to be looking at these multi-hybrid Punnett squares. On an exam, you will be expected to utilize Punnett squares with respect to 1 gene. Before we continue, I want to emphasize that even if an allele for a given trait is the dominant allele, that doesn't necessarily mean it will be the dominant phenotype of the population. For example, in humans, we can look at 2 different traits. Polydacty, which is having extra fingers or extra toes, and Huntington's disease, which is a neurodegenerative disease. Both of these conditions are governed by a dominant allele. So if you inherit at least 1 copy of the dominant allele for polydacty, you will have extra fingers and or extra toes. And if you inherit at least 1 dominant allele of the Huntington disease gene, you will develop Huntington's disease. So both of these examples are examples in which the dominant allele produces a very specific phenotype, but that phenotype is not the predominant phenotype observed in the population. Most of us do not have extra fingers or extra toes, and most of us will not develop Huntington's disease. A test cross can be used to determine the genotype of an organism. For example, here is pictured a tall pea plant. Being tall is the dominant phenotype, so this plant we know has at least 1 copy of the dominant allele. The genotype of this plant could either be homozygous dominant, capital T, or heterozygote, capital T, lowercase t. To figure out what the genotype of this particular plant is, we can cross it with a plant that has the recessive phenotype, a plant that must be homozygous recessive, little t. On the bottom of this slide, we see the 2 Punnett squares. On the left is the Punnett square that represents if the dominant parent plant was homozygous dominant, and we know the other parent plant is homozygous recessive. The result is that the offspring would all be heterozygotes. and would all have the dominant phenotype. On the right-hand side is a Punnett square for if the parent plant was a heterozygote. In here we see that if this was the case, we would see roughly half the plants being heterozygotes and having the dominant phenotype of being tall, and half of the offspring would be of the recessive phenotype and be short. So if you don't know what genotype your parent plant is, you can cross it with a plant that has the recessive phenotype. And if you get all offspring that are of the dominant phenotype, it's likely that your parent plant was a homozygous dominant parent plant. On the other hand, if you perform the cross and you get 50% of the dominant phenotype and 50% of the recessive phenotype, this indicates that your parent plant was heterozygote. Mendel had several important findings, one of which is called principle of segregation. Principle of segregation says that prior to sexual reproduction, the alleles carried by a parent must be separated. This is what is happening in meiosis. We learned about meiosis in the previous chapter, and we learned that meiosis is a type of cell division in which the number of chromosomes is reduced. So we are diploid organisms. We have 2 copies of every gene. When we make our gametes, sperm for men or women for eggs, we are reducing the number of chromosomes in those gametes by half, so that our gametes only have 1 copy of every gene. On slide 12, we look at Mendel's principle of independent assortment. In short, the principle of independent assortment says that the inheritance of one gene does not influence the inheritance of a second gene. So if we look at this slide and we look at this cell right here, I can see that this cell has already replicated its DNA. Remember, DNA replication occurs in interphase, specifically during S phase. So what we see here are sister chromatids, which are identical copies of a chromosome. And you can see that both of these sister chromatids have the recessive lowercase b allele. Here in red are the sister chromatids for the homologous chromosome, which contain the dominant allele, capital B. Here is a second set of homologous chromosomes that have a different gene called gene S. And again, we see the blue sister chromatids happen to have the recessive lowercase s allele, and the red sister chromatids have the uppercase s allele. The way these sister chromatids and homologous chromosomes separate from one another is random. So one possibility is for the recessive alleles to end up in the same gamete. So we see that these 2 gametes produced here both by chance inherited the recessive B and the recessive S alleles. And these 2 gametes, again, just by chance, inherited the dominant allele for B and gene S. Equally likely is the scenario shown on the right, in which the gametes have inherited the lowercase b allele and the capital S allele, and the capital B allele and the recessive lowercase s allele. So again, the principle of independent assortment says that each of these scenarios is equally likely to occur. I will go through the sample genetic problems in a separate video. What we've learned so far is Mendelian inheritance, when there is one gene with two alleles for one trait. However, there are a number of different inheritance patterns that do not follow the simple Mendelian inheritance patterns. We call that non-Mendelian inheritance. One example of non-Mendelian inheritance is linked genes. Linked genes are genes that tend to be inherited together. This is because they are located on the same chromosome. In fact, the closer two genes are on the same chromosome, the more likely they are to be inherited together. Another example of non-Mendelian inheritance is incomplete dominance. In incomplete dominance, the heterozygote individual displays a phenotype in between the homozygous dominant phenotype and the homozygous recessive phenotype. For example, in this flower, the dominant phenotype is red. The recessive phenotype is white. However, when these two plants are crossed, it produces a heterozygote plant with a phenotype in between the two. So red plus white equals pink. This is an example of incomplete dominance. In humans, a great example of incomplete dominance is sickle cell anemia. I will go through this example in a separate video. Codominance is another example of non-Mendelian inheritance. In codominance, the heterozygote individuals express both the dominant and the recessive phenotypes. A great example of this is human blood types. I will present these genetic problems in a separate video. X-linked inheritance is another example of non-Mendelian inheritance. This is because genes that are located on the X chromosome are inherited differently in men versus women, because women have two copies of the X chromosome, while men only have one copy of the X chromosome, as their second sex chromosome is the Y chromosome. Women will have regular dominant recessive expression of genes located on the X chromosome because women have two copies of the X chromosome. However, men will display a different inheritance pattern because they are hemizygous. They only have one copy of the X chromosome. We see this in color blindness, which is governed by a gene on the X chromosome. So a woman could have three possible genotypes. A woman can be homozygous dominant and have normal color vision. A woman can be heterozygote, and because they have one healthy dominant allele, the woman will also have normal color vision. Or a woman could be homozygous recessive, and in that case have two copies of that recessive allele and thus will be colorblind. Men, however, only have one copy of the X chromosome. So whichever allele happens to be on their X chromosome is the phenotype they will express. So if a man inherits a dominant allele, the man will have normal color vision. But if the man inherits that recessive allele and only needs one copy of that recessive allele, he will be colorblind. This can be seen visually in these figures on slide 21. Again, I will go through these genetic problems in a separate video. Because women have 2 copies of the X chromosome, while men only have 1 copy of the X chromosome, there is what we call dosage compensation. Women will silence one of the X chromosomes, called X inactivation. Here we see an image of a cell that has 2 copies of the X chromosome. During development, at random, one of these X chromosomes is inactivated. It essentially gets covered in proteins, which cause the chromosome to become what we call a bar body. And this bar body is in a state that the genes are inaccessible to the cell. So the cell will only use the genes on this X chromosome here in red and not on the X chromosome here in blue that is now a bar body. An example of this is calico cats. So here in this figure on slide 24, we see early on in the embryo, all of these cells contain 2 copies of the X chromosome. So we are talking about a female cat. And we see that 1 copy of the X chromosome here in red has the allele for orange color fur, while the chromosome shown here in blue has the allele for black fur. Again, during development, at random, one of these chromosomes is silenced in every cell. So you see in some of these cells, the blue X chromosome was inactivated. We see these bar bodies here, which means the X chromosome that has the allele for orange color fur is the only one that will be expressed. And so these cells then will express orange fur color. We see in these other cells, however, that it is the blue X chromosome that is being expressed because the red chromosome was inactivated. In these cells, we see that only the allele for black fur is available because, again, that red chromosome has been inactivated. So these cells will express black fur. And this is what accounts for the coloration patterns in calico cats. There are other examples of traits which are influenced by sex, and these are for any genes that are located on the sex chromosomes, either the X or the Y chromosome. Another example of non-Mendelian inheritance is when genes interact with one another. The first we're looking at is called epistasis. In epistasis, 2 genes interact such that one gene controls the expression of a second gene. A great example of this is fur color in Labrador retrievers. So there are 3 different versions of labs. You can have a black lab, a chocolate lab, or a yellow lab. This is because there are 2 different genes at work. One gene dictates what color pigmentation should be deposited in the fur. The dominant allele, capital B, dictates that the black pigment should be deposited into the hair, while the recessive allele dictates that brown pigment should be deposited in the fur. However, this gene is being governed by a second gene called the color deposit gene. This gene, if you inherit the dominant allele, allows the expression of the color coat gene, while the recessive color deposit allele, lowercase e, blocks expression of the color gene. So, for example, these yellow labs here are yellow because they are homozygous recessive for the color deposit gene. So it doesn't matter whether or not they have the genes or the alleles for black or brown fur, because that recessive color deposit gene, little e, is blocking the expression of the coat color gene. Pleiotropy is when one gene has multiple effects. An example of this is cystic fibrosis. Polygenic inheritance is when there is more than one gene that influences one trait. This is quite common in humans, and one example is skin color. There are a number of different genes with many different alleles that govern a single trait, skin color. So you can see that represented here in this figure. There is a distribution of skin tone from lighter to darker, and this is dictated by a number of different genes. There are examples of organisms in which expression of certain genes is influenced by the environment. For example, these are hydrangeas, and the flower color of these hydrangeas is influenced by the pH of the soil. Another example of how the environment can influence expression of genes is the fur color in Siamese cats. That concludes the video of Chapter 11. Thanks for watching. Now go wash your hands.